Can you hear me?
About how we can understand flavivirus replication and pathogenesis through virus-host protein-protein interaction mapping. And what I want to highlight at the beginning of my talk is that my lab at UC Davis is very new. We're less than a year old. And so a lot of the data that I'm going to be showing you today is data that I generated along with others at UCSF during my postdoc in Raul Andino and Nevin Krogan's labs. And these are stories that we're continuing to work on at UC Davis in my own group. Okay, and so my lab focuses right now on mosquito-borne flaviviruses, and that's partly because they're really interesting viruses, but also because they have a really outsized impact on public health. So if we look at mosquito-borne flaviviruses in general, these include dengue virus, West Nile virus, yellow fever virus, and Zika virus, to name a few. Over 40% of the world's population is at risk of infection by one of these mosquito-borne flaviviruses. And to give you some hard numbers on the global burden of some of these diseases, dengue virus is responsible for nearly 400 million infections annually. Now, most of these infections are asymptomatic, about 75% are asymptomatic, but when we do see disease, it can be a mild disease like a rash or a fever in the case of dengue, or more severe disease like dengue shock syndrome, which is particularly prevalent in smaller children. And then more recently, with the emergence of Zika virus, we've learned that these diseases are not only a risk to adults and children, but also to fetuses that are infected in utero. And so this is an image of an infant that was infected in utero with Zika virus and suffers from congenital Zika syndrome and severe microcephaly. And so because of this impact on human health, there's an urgent need to really understand how these viruses replicate and how they cause disease as a result. And so we try to answer that question by understanding how these flaviviruses hijack host cellular pathways. And the reason for this is highlighted in this image here, where we're looking at the basic replication cycle of flaviviruses. And so in the most basic sense, these viruses have to enter the cell, replicate its genome, package these genomes into new daughter virions, and then exit the cell. And to accomplish this, viruses often rely on cellular machinery to do all these tasks. And it's often done through physical interactions between viral proteins and host proteins. And so we use mass spectrometry and global proteomics to identify these physical interactions between a viral protein, which I'm showing as a square, and these host proteins, which I'm showing as circles. And that's a convention I'm going to use throughout the rest of this talk. And so we use these physical connections as a starting point to understand how these viruses replicate and potentially how, by interacting with these different pathways, dysregulate those pathways and cause disease. And so today, I'm going to tell you about, first, an overview of the methodology we use to develop these protein-protein interaction maps. And then I'm going to tell you two stories that came out of the data regarding flavivirus host protein interactions. One is an immune evasion mechanism, and the second is a molecular mechanism for neuropathogenesis that we think is playing an important role for Zika virus. Okay, so since we're going to be looking at physical interactions between flavivirus proteins and human proteins, I want to first introduce you to the flavivirus polyprotein. So flaviviruses are positive sense single-stranded RNA viruses. They're translated as one long polyprotein that's then cleaved into distinct protein products that are indicated by these cleavage points with arrows. And so the proteins that I want to highlight for the purposes of this talk are NS4A, which is this orange protein here. Its primary role is to bend the ER membrane to create these invaginations where virus replication occurs. And then the second protein I want to highlight is NS5, right here on the C terminus of the polyprotein. And that is the viral methyltransferase and polymerase. And it's responsible for replicating these viral genomes in these replication compartments. Okay. So to map all of these virus-host protein-protein interactions, we use what is now a standardized affinity purification mass spectrometry pipeline that the Krogan lab has really spearheaded. So this is well established in the Krogan lab. And what we did is, when I arrived as a postdoc, we were working on dengue virus, and so we cloned all of the dengue viral open reading frames into a plasmid containing a C-terminal strep affinity tag, expressed these in HEC293 T cells, purified the viral proteins, and then analyzed those samples by mass spectrometry to identify any of the human proteins co-purifying with the viral proteins. And so if you've done any mass spec experiments in the recent past, what you know is that these machines are incredibly sensitive and detect hundreds, maybe thousands of proteins in a single protein purification sample. And at the very top of your raw data of mass spec that comes back from the core facility are your usual promiscuous, sort of less interesting and highly abundant proteins like actin, tubulin, and ribosomal proteins. So very important for cell biology, but maybe not relevant for virus replication and pathogenesis. And so we need a way of sifting through all that data. And what the Krogan lab has established with several viruses now at this point is that the specificity of the virus-host protein-protein interaction is really key to identifying biologically relevant interactions that have some sort of physical meaning for the virus and maybe for a disease. And so in terms of specificity, what I mean is that the human protein interacts specifically with one or maybe two viral proteins at most out of the 10 dengue virus proteins that we started with. And so by doing this systematically for each dengue virus protein, we have all of these internal controls to identify that specificity really critically. And this has been formalized in a scoring system that the Krogan lab has developed, which is called MIST. And so using this, we can go from, say, 13,000 protein-protein interactions for all 10 dengue virus proteins down to a smaller set of about 200 protein interactions that we think are more interesting based on that specificity. And so we did this not only for dengue virus, but also for Zika virus, because after we finished this for dengue, Zika had just emerged in 2016, and it was a really interesting way to compare these two virus-host protein-protein interaction networks. And our hope was that by comparing these two networks, we could identify things that were common between the two viruses that may reflect their shared biology and replication. But then by digging more deeply into the Zika virus data set, we might be able to find physical interactions that explain its neuropathogenesis. And so what I'm going to tell you about is one of these shared interactions that relates to a common immune evasion mechanism, we believe, and then one interaction from the Zika data set that relates to its neuropathogenesis. Okay, so in comparing these two networks, we can look at a protein by protein comparison, but that very quickly gets overwhelming. And so you can also take a higher level view of it, looking at gene ontology enrichment categories. And so that's what I'm showing you here on this graph. And so here you're looking at a subset of the data for both dengue and Zika virus, where each square here in the center is a viral protein from both data sets. And then the circles here represent gene ontology enrichment categories that were either enriched in the dengue data set if it's blue or the Zika data set if it's pink. And so what I hope you can gather from this slide is not that you can read every single Go enrichment category in its small text, but to observe that NS5, the viral polymerase, really has a lot of shared enrichment categories for both data sets. And specifically, enrichment categories relating to gene expression. And what we saw were enrichments for proteins related to chromatin modification, transcription, splicing, and RNA processing. And what struck us the most was this interaction between the viral polymerase NS5 and this protein complex PAF1C. So here we're looking at the specific physical interactions that we identified by mass spec that contribute to the Go enrichment category in this data set. So what we identified were 4 proteins from PAF1C that interacted with dengue virus NS5, and 2 of those proteins that also interacted with high confidence with Zika NS5. And so this is one of the interactions. Because of this strong signal, we decided to pursue further. And the first question we asked was, well, how does PAF1C impact virus replication? And so this is work that was done with Holly Ramage, who was a postdoc actually in Melanie Ott's lab, which is how I got to know her, and she's now at UPenn. She performed RNA interference knockdown of PAF1C complex members, 3 different complex members here, and then infected with either dengue or Zika virus to determine the impact on virus replication, or infectivity, actually. And so what you can observe here, if you look at the blue bars compared to the black bar, which is a control, is that knockdown of 3 of these PAF1C complex members increases viral infectivity for both dengue and Zika. And so this suggests that PAF1C is somehow restricting virus replication for flaviviruses, but we wanted to understand whether this physical interaction with NS5 has anything to do with its biology. And so to answer that question, we first need to understand what PAF1C's role is in the cell. So I said it's involved in chromatin modification and transcriptional regulation. And specifically, what it's responsible for is regulating transcriptional elongation. And so this is an abridged version of what you need to know. There are a lot of details I'm leaving out. But what we know about PAF1C is, first of all, it's responsible for regulating transcriptional elongation at a lot of different genes. It's broadly distributed across the genome if you do chip analysis on it. But in regards to viral infection, what we know is that it's actively recruited to interferon-stimulated genes, or ISGs, to facilitate their transcriptional elongation. And so what happens during an immune stimulation is that PAF1C is recruited to the promoter of these ISGs, where it serves as a scaffold to recruit other chromatin-modifying enzymes and create a chromatin environment that's conducive to transcriptional elongation. And this allows RNA Pol2 to release and fully elongate its transcripts. It's also recruited to the 3' end of these gene bodies, where it facilitates 3' end processing of the fully elongated transcripts. And so what we wanted to know is whether NS5 in any way inhibits PAF1C recruitment to these ISGs. And so to answer this question, I performed the following experiment. I created cells that either express GFP or NS5 from dengue virus in this case. And then I stimulated with interferon beta to mimic a viral infection and performed ChIP-seq on PAF1C for all of these different conditions. And so then what we did was we looked at PAF1C occupancy at ISGs that are relevant for dengue virus replication. And so this is a subset of 67 ISGs. And for the GFP expressing cells, what you can see is that in the presence of interferon, we see a nice increase in the occupancy of PAF1C at these genes. Whereas in cells expressing NS5, we don't see that behavior. So NS5 expression in these cells inhibits PAF1C recruitment to ISGs. And what's interesting is that this inhibition appears to be specific to ISGs. So when we look at other genes in the genome, we don't see that sort of inhibition of PAF1C recruitment. We also don't see any increase in PAF1C occupancy in the GFP cells, which is reassuring. And so what this suggests in terms of our model is that NS5 is inhibiting PAF1C recruitment to ISGs in some specific way. And so some of the questions that we're looking at now at UC Davis is, first of all, how conserved is this antagonism? Does it extend to other mosquito-borne and potentially tick-borne flaviviruses? What are the viral determinants of this interaction? And can we show that based on those viral determinants, the physical interaction is required for this antagonism? And then finally, because this antagonism appears to be specific, what are the genes that are regulating PAF1C recruitment to ISGs? And is NS5 perturbing that recruitment in particular? And so some of these questions are being answered by a postdoc that joined the lab, Maureen Petit, who's here today. And she's done a heroic amount of work, not only in setting up the lab, but also building some of the tools related to this project. Okay, so now I'm going to tell you about how we dug into the Zika virus data set to really understand whether any of these physical interactions might explain the neuropathogenesis associated with Zika virus. And so in the PAF1C example, I showed you how gene ontology enrichment analysis can help you identify some of the physical interactions that are most interesting. In this story, it was really a brute force approach of reading about hundreds of proteins in the list of our top hits and determining whether any of them had any relevance to neurodevelopment or brain disease. And so in reading about all of these proteins, one interaction really stood out to me, and that was an interaction between Zika virus NS4A protein, that's that membrane remodeler that I mentioned earlier, and ANKL2, which is a human protein. And there's not much literature on ANKL2, but one paper that got me really excited was this paper that was published a few years ago by Hugo Bellin's lab at Baylor College of Medicine. And they performed a Drosophila mutagenesis screen looking for neurodevelopment phenotypes. And they overlaid this mutagenesis screen with an exome sequencing database of patients that had their exomes sequenced because they suffer from rare Mendelian disorders. And it was basically a way for them to try and figure out what the genes responsible for those disorders might be. And so when they overlaid these two data sets, there was a few genes that they found interesting, but one was ANKL2. So they had these mutant flies that had a bristle phenotype, so they had fewer bristles on their thorax. And then they had human individuals with ANKL2 mutations in this database. And what's striking about these individuals is that, well, this individual suffers from severe microcephaly. And so this patient has 4 mutations. One of the mutations is in the ANKL2 gene, and both alleles are mutated. One is a truncation mutant, and the other is a point mutant. And so based on this patient, the Bellin group went back to their flies to determine whether their flies also had small brains, because all they had was this bristle phenotype initially. And so they quantified the brain volume of these flies, and what they found is that the ANKL2 mutants had significantly reduced brain volumes, and what was nice is that they could rescue this phenotype by expressing wild-type human ANKL2. So now they have this system to test human ANKL2 function in flies. And so based on these data, we contacted Hugo Bellin's group to ask them if they would test whether Zika NS4A also created a small brain phenotype in flies. And so this is all work that was done by Nicole Link, who's a postdoc in the Bellin group. And she's really done a heroic amount of quantification of fly brains. And so what she did was she first looked at brain volume of a control fly, which is expressing some unrelated protein, which she found does not affect brain volume, CD8-GFP. And then when she expressed Zika NS4A, she found a significant decrease in brain volume. And what was nice is that she was able to rescue this phenotype with human wild-type ANKL2 expression. And then what was really beautiful about all of these fly genetics experiments is that she could also test the mutant ANKL2 allele that was identified in that original patient that suffered from hereditary microcephaly. And that's the Q782X mutation. And that version of ANKL2 cannot rescue the brain phenotype for these Zika virus NS4A flies. And so this is really exciting. And so what's also really great about Nicole is that over the course of her postdoc, she's learned a lot about ANKL2 biology independent of Zika virus. And so what she's learned is that in neuroprogenitor cells in flies, ANKL2 has 3 main functions. The first is positively regulating cell division and progenitor cell, and then the second is positively regulating progenitor cell regulation and the asymmetric stem cell division that's required for some of these neuroprogenitor cells in the fly brain. And then it also positively regulates cell survival to prevent cell death. And so the mutation in ANKL2 in her ANKL2 mutant flies impacts all three of these aspects of ANKL2 function. And so she has all of these assays to test for downstream ANKL2 function already set up. And so she started looking at whether any of these functions were impacted by Zika virus NS4A. And so for the purpose of time, I'm not going to talk about the impacts on cell division and progenitor cell regulation, but what I will say is that there is a mild phenotype if we use the right genetic background. But what I am going to tell you about is some of the cell survival data, which is more compelling at this point. And so what Nicole performed is a tunnel assay to quantify cell death, and here she's looking at cell death normalized to the brain volume, since the brain volume is different in flies. And so for the control, she saw a minimal amount of cell death that was significantly increased when the flies expressed Zika NS4A, and that cell death could be rescued by expression of human ANKL2. And so our current working model is that ANKL2 has these impacts on cell division, cell survival, and progenitor cell regulation, and that Zika virus NS4A inhibits ANKL2 function, we believe, through this physical interaction, primarily through the cell survival pathway, but the other pathways also appear to be affected to some degree. And so moving forward, we have a couple different questions. First, on the virology side, what role does ANKL2 play in Zika virus replication? Is it interacting with it to facilitate replication in some way? And is this phenotype more pronounced in the tissue that Zika virus tends to affect and be pathogenic in, which is neuroprogenitor cells? And so that's something that we're working on using CRISPR knockout. And then on the more systems biology side and fundamental cell biology side, how do ANKL2 interactions change during these two disease states? Because now we have two really nice tools to use. We have a hereditary disease state and an infectious disease state that presumably perturb the same pathway, and we're hoping through the same changes in the physical interactions that mediate these downstream effects. And so we'll be performing affinity purification and mass spec on ANKL2 in these two disease states in neuroprogenitor cells to tackle some of those questions. Okay, so with that, I'd like to acknowledge all of the people that were involved in the work that I showed you today, in particular, Nevin Krogan and Raul Anduno for their mentorship over the last several years, and the many members of their lab that were involved in generating the massive amount of proteomic and virus data that I showed you. Our collaborators, Holly Ramage, Nicole Link, and Hugo Bellin, the members of the Shah lab, and the funding sources that supported my postdoctoral work. And I'd be happy to take any questions.
The past concepts quite a lot of cells, and the main kinds of mostly gets included to interact with RNA two. There are two related questions. Number one is NS5 protein accumulated sufficient high level in the nucleus. Number two, whether it actually works by breaking up interaction between limits and path concepts.
So to repeat the question, there are two parts. The first is whether NS5 actually accumulates in the nucleus to sufficient levels to perturb interactions with PAF1C, which is obviously associated with chromatin and in the nucleus. And to answer that question first, yes, NS5 is definitely in the nucleus. So this is one of the really interesting things about flaviviruses. So they're plus-strand RNA viruses. Their replication should theoretically be exclusively cytoplasmic. And as such, the polymerase, which is the main enzyme responsible for genome replication, should be exclusively cytoplasmic. But flavivirus NS5s, depending on which ones you look at, spend a significant amount of their time in the nucleus. And we believe that part of that reason is to inhibit the innate immune response. And so the second question was whether NS5 actually breaks the interaction between PAF1C and RNA Pol2. That is something that I had not considered, but it would, I guess if that were the case, then I would assume that we would see broader effects that were not just specific to ISGs, depending on how often PAF1C is turned over and leaving RNA Pol2. And we haven't observed that, but it's certainly something that we could look at. Yeah, thank you for the suggestion.
A related question. I was wondering, because most of the things that NS5 is interacting with are not things that the virus would need, splicing factors or transcription factors, you might expect that you'd see sort of broad-scale changes in post-RNA biogenesis or processing in the context of flavivirus infection. I wonder if you look at that. And then the other question is, to what extent do you think that it's interacting with these in the nucleus as opposed to maybe preventing their nuclear entry, and you see relocalization of those proteins and maybe association with mitochondria like that?
Yeah, okay, so again, two-part question. The first part was whether we see any broad-scale changes in RNA processing because of all of these interactions with, different types of RNA processing machinery and transcription regulators. And this is work that we have done a little bit with Andrea Gramarnik's group at the Leloir Institute in Argentina. And they sort of looked at this physical interaction between NS5 and the spliceosome. And we performed some RNA-seq experiments with them. And they do see overall changes, for example, in splicing patterns. And they do see changes in splicing of innate immune effectors, which would help the virus replicate better. But we haven't looked at any of these related to, say, PAF1C in particular, and whether 3' end processing is changing in particular. And so I forgot the second part of your question. Oh, yes, yeah. And so the second question was whether whether NS5 is interacting with PAF1C in the nucleus, or if it's preventing localization and import into the nucleus of PAF1C. That's not something that I have looked at directly in terms of looking for co-localization of PAF1C with NS5. What I will say is that NS5, in the majority of cells that I've looked at by immunofluorescence, it's predominantly nuclear. And so identifying cells where there is NS5 that's cytoplasmic and not nuclear is the minority of cases. And so then we would just have to do the localization with PAF1C. Yeah, we should look.
Okay, last question.
Yeah.
So my first question is, you presented a model of where specific interactions are part of the same pathway. I was wondering if the part of the same pathway that leads to somehow giving rise to gene size, microcephaly, or decreased brain size. So I was wondering how you differentiate between that model, a model where both of these things are specifically in this cell type through different mechanisms. So it could be that NFO2 somehow is cytotoxic for one reason, and NS4 is virus that's in the cells. And you have the microcephaly, which is the expression or the localization of both of those proteins in neuroprogenitors. Following up on that, why is the neural system specifically affected? Why is brain size affected versus other things? Because the virus should affect all the cells. So is it a localization effect that accounts for a specific effect on brain size?
Okay, so the first question is, could it be that NS4A and ANCL2 mutation are actually impacting brain development independently and not through the same pathway? That could potentially be true, and it's certainly not mutually exclusive. It could be that NS4A is impacting brain development through ANCL2 and through something else, and there's literature out there that suggests that maybe NS4A has other impacts on neuroprogenitor division, independent of ANCL2. But what I would say is that the rescue in flies by human ANCL2 really suggests that it's perturbing ANCL2 function, specifically in our system. And then the second question regarding, is it something specific about the brain that Zika virus is impacting to cause the neuropathogenesis? What I will say is that the details that I didn't talk about are that the Zika NS4A drivers that Nicole used were ubiquitously expressed throughout the fly. So they weren't specific to the brain or to any specific neuro tissue. And so those are experiments that she's currently doing right now to see if there are specific tissues in the brain that are more severely impacted